Hello again. Here we are with Chapter 12, Part 1, and this is the B part of Part 1. So this would have been the first lecture we'd had if we'd had normal classes. And we're following up on muscles. This time we're going to get into the mechanisms of contraction and some of the theory behind how muscles shorten. Believe it or not, it was not readily ascertainable as to how muscles actually shorten. And let me give you the visual example for you. If you take a piece of bacon, for instance, and you put it into a frying pan, you might realize that you put that bacon in the frying pan and sometimes it's so long it can't even fit in the frying pan. But by the time you're done cooking it, it's really short. It's half the length that it was previous. And that gets to the idea of the two competing theories of muscle contraction. So we're going to show you some of the evidence for why we have what's called the sliding filament theory. And we're also going to go into the mechanisms of how muscles generate force. It's a very, very important concept, and it's called the Powerstroke. So let's move on. Before we can get into this idea of sliding filament theory, we need to understand the filaments and understand the morphology that we see that gives rise to the sliding filament theory. So we have different bands. You have actin and myosin are the two most prevalent proteins, and they are in sort of alternating order. So we have a band here that's primarily actin, and then we have a band here that's a little bit of overlap, but also myosin. We have this disc-like structure. We have a disc-like structure here. It's hard to see in this picture, but that's what we have. We have this alternating regimen or regimented structure, regimental-like structure. What I mean by regimental is that let's say you go to a military cemetery, or if you see a military unit on parade, they're evenly spaced. They have very clear ranks. You can see down, you can see the discipline in the patterning, and that's what we mean by regimented. So we have these different types of bands and discs, or sometimes they call them lines. In any case, A band is the band that is occupied by the protein myosin. So right here in this purple, this area here that we see from one end of the myosin to the other would be the A band. And you can see that it's not only just myosin. From time to time we have overlap with actin and myosin, or sometimes we have this thing called an M line, where the myosins are actually attached to each other through these cross-linking proteins. I'll show you some more pictures of that in a little bit. But the A band is the myosin band. In the middle of that A band, we have the M line. Again, that's where those cross-linker proteins are. And we even have an interior zone where there is no overlap with actin. So right here we have overlap with actin, right here we have overlap with actin, but right in this area there's no overlap with actin, and that's called the H zone. So we have the A band, we have the M line, and then we have the H zone. And you should probably draw this stuff. I wouldn't oppose you to drawing this stuff out and working with it a little bit so you understand it better. On the actin side, we have what's called the I band. I don't know why they don't call it the I zone or something else, but in any case, it's I band. I band is shown, half of it is shown here, and another half of that I band is shown here. So if I were able to duplicate this, I'd have another myosin over here. You could see the extent of that I band. I band is where we do not have overlapping actin and myosin. All we have is actin. And in the middle of that I band, we have the Z disc. Again, the Z disc is like the M line, only the Z disc is cross-linkers between the microfilaments of actin. We also define this sarcomere, that is to say the contractile unit of muscle, as running from Z disc to Z disc. So this would be one sarcomere right here, and we would see another sarcomere right here. We should see a Z disc over here. In any case, that's the definition of a sarcomere. So M for myosin, Z, what happens when you get to Z, the next letter, if you start over on the alphabet, you get back to A. So we have actin. And again, we have this myosin, and it's complex. We're going to break this out a little bit. We actually have two different domains here. We have this sort of stalk domain of myosin and these little catalytic heads. And the thin filaments are multiple bands of actin together. I'll talk about Titan and Nebula a little bit later. We have roles for those. So here's that myosin. Again, here's this tail, this stalk-like tail that's interacting with other tails. Here are the cross-linker proteins for the M line. Here are the cross-linker proteins for the Z disc. You can also see in the actin microfilament, the thin filament, that we have a helical-like structure that not only has actin, but it also has two proteins, one called troponin, which is a calcium sensing protein, and another protein called tropomyosin, which regulates the ability of the myosin head to interact with actin. You see these little yellow areas on actin? Those little yellow areas are high affinity sites for myosin. And right now in this situation, the tropomyosin is covering that high affinity site, so the myosin head cannot bind tightly. But again, these are stories that we're going to talk about today. This is a relatively intense set of slides in this lecture. So there's that H zone. Again, the H zone is the non-overlapping part of the A band. Here's the I band. Again, the non-overlapping part of the actin in the sarcomere. And so now we have a histological slide showing you the A band with the H zone in the middle. Here is the M line. Here is the I band for actin with a Z disc, or they say Z line in this picture. Okay. So let's start putting this into the context of a muscle fiber / muscle cell. So here's our sarcomere unit here, and these things are attached serially, so we see long strands of these things. So individual myofibrils, which are serial strands of sarcomeres, are all stacked together inside the muscle fiber like logs on a firewood pile. Also surrounding these myofibrils are sarcoplasmic reticulum. I mentioned this in the last lecture that sarcoplasmic reticulum serves a very, very specialized role, an important role in muscle contraction. They store calcium and then they release it and then reuptake it selectively. We also have this very special structure, which is basically an extension of the plasma membrane called a T-tubule. The T-tubule penetrates down all the way through the muscle fiber so that what you get is a continuous and synchronous contraction of the myofibrils and the sarcomeres in those myofibrils. If you don't have synchronous contraction of the myofibrils, what you'll get is a deviation from the center line, and that can be exceptionally painful or overly destructive of the muscle fibers. You can actually rupture muscle fibers. And this is very, very common in overuse. You get asymmetric contraction, and that sort of thing leads to rupture of the muscle cells, and that leads to pain and loss of function and so forth. So when you're looking at this picture, we have all of these terms here. You need to be asking yourself, what do these things do? Again, a sort of a haiku definition of what each and everything in these pictures represents and what their basic functions are. What do these things do? What is a myofibril? What is a thick filament? What is a thin filament? And so forth and so forth. I mentioned this a couple of slides earlier, but again, this is just showing you these T-tubules as they penetrate down into the cytosol in the central part of the cell. The point is, for a T-tubule is to spread action potentials down in all the way through the cytosol of a muscle fiber so that you get synchronous contraction of all of the sarcomeres. Skeletal muscle fibers are very, very synchronized. All of the sarcomeres will fire in a muscle fiber simultaneously. How we vary strength of contraction in muscle fibers or muscles is how many fibers we fire simultaneously. And then there's another factor called the length-tension relationship that we'll get to a little bit, not today, but we'll get to that later. So there are two ways that we vary strength of contraction in muscles. But we never vary the number of sarcomeres that we fire in any given muscle fiber. They all should fire simultaneously. And that's because we have these T-tubules. I'm not going to worry about the triad all that much or the names of the different parts of the sarcoplasmic reticulum. What you have is a very, very intimate association of T-tubules and plasma membrane with the sarcoplasmic reticulum so that you have excitation-contraction coupling. That we get an action potential that spreads all around and through the muscle fiber. That causes release of calcium, and then that leads to contraction of the sarcomere. So the T-tubule will bring action potentials down into the interior of the muscle fiber for a synchronous contraction of all the sarcomeres. Sarcoplasmic reticulum stores and reuptakes calcium. It's a very, very important regulator of cytosolic calcium. We release calcium, triggers contraction. We pull calcium back in order to affect relaxation. A little bit of reinforcement on the structures that we find with myosin. So we have this tail structure, we have this hinge region, and then we have these catalytic myosin heads. Now I want you to think back to early in the semester when we talked about motor proteins and I showed you that video. That's what's going on with these myosin heads. They're rotating or they're moving back and forth from a cocked position, cocked being this way to the right, and a flexed position to the left. So when you think flex, you flex your arm, you're bending your elbow. That's what's going on with these myosin heads. We have this tail structure that allows for myosin proteins to bind together and allows them to cross-link in the case of the M line. That provides a firm structure onto which we can impart force. The catalytic head, on the other hand, I like to use the analogy of a cat's paw. The catalytic head not only hydrolyzes ATP and can move from a cocked position, that is, to the right, I would love to say extended, or a flexed position to the left. And so this cat's paw right here is in a flexed position. So they can swivel back and forth on these hinge regions, and they use ATP to do that. I want you to get this down right now. I'd love it if you would write this down. We do not cause flexing with ATP. So when we're generating force, the energy has already been released by ATP. We are not flexing because of ATP. We are cocking. We are extending the myosin head to the right with ATP. We're not doing the flex to the left, we're doing the extension to the right. That's counterintuitive, and a lot of students will not realize that and miss some easy points. So there's that myosin head. It's catalytic. It hydrolyzes ATP in order to move from a cocked position or move into a cocked position. And then when we release the myosin, when it acts with actin, it doesn't hydrolyze ATP at that time, but it does flex. So we're putting potential energy into the myosin by cocking it, and then we're releasing that potential energy during the flex. I'm going to come back to that a little bit later, but I wanted to get started early on that counterintuitive aspect of ATP hydrolysis in myosin. And so a little bit again about the actin thin filament. Here it says G-actin. G-actin is a single monomer of the longer filamentous actin. G-actin is like a nucleotide in DNA, so it's an actin monomer, whereas F-actin for filamentous actin includes all of these things. So we've got a couple of strands of F-actin. We've got troponin and tropomyosin. Again, we'll talk about those a little bit later. They regulate the ability of myosin and actin to interact. And then now we have a protein here called nebulin. They call it nebulin because they didn't know what it did, but now we know what it does, or we think we know what it does. It is an alignment protein. It helps to keep the filamentous actin in a nice, straight, rod-like configuration. If we didn't have the nebulin there, what you might see is a bending of the filamentous actin, and we don't want that. I'll hang on to titin for a little bit later. But to kind of summarize, actin is a filamentous structure on which myosin pulls. Myosin is doing the pulling, as far as we know, in the sliding filament theory. So actin is actually serving a sort of relatively stationary process, or at least it stays straight and is what myosin pulls against. Troponin and tropomyosin regulate the ability of myosin to bind tightly to actin. When the troponin and tropomyosin are covering that sort of yellow high affinity patch, the myosin can still bind to the actin, it's just that it can't bind very strongly. Let me give you a personal example. Let's say you just take your hand and make your wrist limp and move it around. You know, when you have that limp sort of flaccid muscle, the actin and the myosin are still interacting. It's just that they're not interacting tightly. Okay? So if you make your wrist rigid, what you have going on now is you have myosin tightly interacting with actin, and that's why. And of course, myosin is also generating force. But that's what contributes to the rigidity, the tight binding of myosin to actin on that high affinity site, coupled with the power stroke of the myosin. Lastly, titin and nebulin. Nebulin is an alignment, and I guess I'll give away the secret now. Titin is an elastic protein. That allows you to do things like isometric contraction so that your muscles don't rip themselves apart if the appendage doesn't move. So titin is called titin because it was a very, very large protein. It's one of the largest proteins we know of. And it is an elastic element. That is to say, it stretches when sarcomeres are contracting and helps absorb that kinetic energy in a contracting sarcomere. So start putting this all together. We've got our A band with the M line. We've got the H zone. Okay. The H zone can actually change in size as actin and myosin increasingly overlap. The interaction of myosin with actin is collectively known as a cross bridge. So if one myosin head is binding to actin, it is cross bridged to that actin. And so we have multiple cross bridges going on. And these cross bridges can be either tight or they can be loose. Again, here are the pieces of the puzzle. This is a great practice for you. You can, you can erase the terms and then fill in the blanks, that sort of thing. A great practice for the exam. Now, this slide is helping us transition and should hopefully help you visualize the sliding filament theory of protein, of the sarcomere. So what we have here are the filaments, and we have the discs, and we have the general picture that you've seen before of the different overlap from the side. We're going to try to make this a little more three-dimensional, and I'm going to move left to right. So let's start with the I band. The I band is non-overlapping actin. And so the I band, all you see, if you were to cut a piece out of the I band, all you would see are thin filaments. So these little red dots in here are all just thin filaments. So if we took a piece, this little circular thing is a slice out of that, and then what you see when you look at it crosswise, you see is only actin. If we go to the H zone, we go to the H zone and we take a piece out of the H zone. Remember that the H zone is non-overlapping. Now we're not talking about the M line. We'll come to that a little bit later. The H zone is non-overlapping myosin, and there we have our thick filaments. You can see the size of the filaments, thin filaments, thick filaments. If we go to the M line, we would see thick filaments, but we would also see cross-hatching. That's from those accessory proteins that link all of the tails of the myosin proteins together. Lastly, I have a non-I band, non-H zone. This is still part of the A band. Remember that the A band is defined by the myosin. This is still in the A band, but it is not in the H zone. And what do we see? Overlapping actin and myosin. Again, very, very organized, very, very regimented. The only thing we don't see on here, the only slice I don't have is in the Z disc, but you can sort of see it right here. The Z disc would look a lot like the M line for myosin, only instead what you would have are red dots with cross-linker proteins. Again, very, very regimented. So make sure you get this down. I almost certainly will have a question like this on the exam. Again, to understand the sliding filament theory. The sarcomere needs to contract uniformly and synchronously in a piston-like motion. If it deviates from that, it could lead to cell rupture and certainly pain and inflammation. So here's titin and nebulin. Here's our slide about titin and nebulin. Nebulin acts as an alignment protein. It aligns the actin and keeps it nice and straight. So far, we haven't found anything that's nebulin-like in the M line, at least to my knowledge anyway. Maybe those tails in myosin are thick enough that they don't need assistance. Here's our Z disc, that's the actin one. Here's our M line and so forth. And now you can see titin, and titin spans from Z disc to Z disc. It actually stretches when we contract our sarcomeres. It's an elastic element. Yeah, maybe it stabilizes myosin a little bit well, but the role that I want you to focus on is elasticity. So as sarcomeres contract, the titin stretches in order to absorb that shortening so that you aren't ripping other muscular structures. You aren't tearing membrane, that sort of a thing. So titin spans from one Z disc to the M line and serves as an elastic element. Nebulin serves as an alignment element in actin. So a few terms associated with contraction that we need to make sure that we all know. When we create tension, that's because muscles do not push, they pull. If there is a push force, it is because we're pulling across a fulcrum and a fixed point. So tension is the force created by the muscle. Load is the force or the weight opposing that contraction. For instance, even in an isometric contraction, when you're pushing against an object that cannot be moved, that force opposing the contraction is known as load. So we're creating tension in the muscle. Okay? When we contract, we create tension and we move that force against a load. Okay? Relaxation is the release of tension. That is to say, in the sarcomere, in the muscle, you no longer have a shortening effect going on inside those muscle fibers. And it is primarily believed to be of the sliding filament theory. And I've already discussed this idea that instead of the myosin shortening, there were two theories. There was the myosin shortening theory, that myosin and actin were attached, and that myosin got shorter. Pulling on the actin was one theory, and the sliding filament theory was the other. And here was sort of the nail in the coffin. So if we look at a relaxed sarcomere, we have this beautiful I band with a Z disc, and here's a beautiful A band here, and an H zone where you don't see overlap, and an M line. Well, the question is, and I would ask you this on an exam, is how do we know it's sliding and not sarcomere and myosin shortening? Well, if myosin shortening was occurring, what we would see is that this I band wouldn't change in any length. The H zone may or may not change length, but what we would see is that the A band would shorten as a whole. If the myosin was shortening, what we should see is a shortening of the A band and a not shortening of the I band. But what do we see when we contract? what we see is that the A band is identical. We have lost the H zone. This is a contracted sarcomere, and this is an relaxed one. You see there's no more H zone. The A band is completely overlapped with actin, and the I band has gotten shorter. You see that the Z discs have come closer together. So here's a Z disc moving this way and so forth. You see a loss of the H zone. Now the H zone again is not overlapped actin and myosin, and you see a shortening of the I band. So what's happening is these I bands are sliding into the A bands, and that's where we get the sliding filament theory rather than the myosin shortening. If it was myosin shortening, you would see a definite shortening of the A band and no change or little change in the I band. We're going to now move on to the overarching view of events of muscle contraction, just here for a second. So we've talked about the sliding filament theory. Now what we're going to do is show you this contraction relaxation cycle. In the larger scope, what we're going to talk about overall in this chapter is we're going to see some neuromuscular junction events leading to an action potential. That action potential elicits a second message that then causes an effect, the contraction relaxation cycle. That involves the sliding filaments, and it also involves a phenomenon known as a twitch. That is, if you have a single action potential, a release of calcium, you're going to get a contraction and then a relaxation. Later on, in subsequent videos, we're going to talk about this stuff, or we'll work our way upstream this time rather than downstream like we did in the neurons. But for now, what we're going to talk about is the sliding filaments and this generation of force. We're going to talk about the effect first, and then move our way upstream in this chapter. So here is our sarcomere. Again, we have a sliding filament going on. The H zone all but disappears when we contract that sarcomere. The A band, however, stays nice and constant. The Z discs are moving closer together, which infers that the actin is sliding into the myosin. The actin is increasingly overlapping. If it was a myosin shortening, what you would see is the A band would shorten and the I band would not. So how do we get to this force generation? How do we get to this force generation? And that involves a second message. That is the release of calcium into the cytosol of the muscle fiber. That calcium interacts with a protein called troponin, and troponin is a calcium sensor. Troponin, when it binds to the calcium, changes shape. And when it changes shape, it imparts force on the tropomyosin, and so the tropomyosin also moves. So here in the relaxed sarcomere, what we see is that the tropomyosin is overlapping that black high affinity binding site for myosin. I say high affinity binding site because myosin and actin can and do interact. It's just that they can interact weakly or they can interact strongly. Sometimes these pictures are a little bit confusing that way, and I want you to understand that when we're relaxed, actin and myosin are still interacting but weakly. So we get this calcium release, the second message that binds to troponin. Troponin changes shape. It causes tropomyosin to move away from that high affinity site, and now the high affinity site is available for myosin. When that high affinity site is available, we now enter what's called the Powerstroke / contraction relaxation cycle. Now this is the figure that you see in Silverthorne, but I want you to understand that previous slide we had was actually showing this down here. So when I go through this, and I'm going to go through this very, very carefully, take detailed notes. Take detailed notes. There are 3 questions you have to answer about each and every one of these panels. I'm going to start right here. This is the relaxed state. This square right here is known as the relaxed state. So the questions you're going to have to answer. The first question is called the configuration of myosin. Is it cocked? Another word for cocked is extended, but we're going to use the proper terms here. So when we cock, what we're doing is we are extending. So use your arm to do this. Think of the hinge region of myosin as your elbow, your bicep and your shoulder as the tail, and your hand and your lower arm as the actin, the catalytic domain of actin. And so what we do is we go into a cocked position, which is relatively straight, or we go to a flexed position, which is relatively bent. So that's one of the questions you're going to answer for any one of these panels. The next question you need to ask yourself and be able to answer is, how is the actin and myosin, how are the actin and myosin binding? Is that affiliation with actin and myosin loose, or is that affiliation with actin and myosin tight? The third question is the status of ATP, or the power molecule. Sometimes we're going to have ATP bound. You see there's ATP here. Sometimes we're going to have ADP, like for instance right here. The phosphate has left and all we have is ADP right here in the lower left corner. Sometimes we have both, like in the case right here, we have ADP and we have inorganic phosphate. This is not ATP. This is hydrolyzed ATP. That is to say, the ATP has been separated from that inorganic phosphate. Yeah, they're right next to each other, but they are not bound. So this question for this box would be ADP plus PI. Or do we have nothing? Is there no nucleotide there? For instance, the ADP is left here, and this is now empty. We're empty. Okay? We're empty. So you need to be able to answer this. And now what I've done is I've put the question and the possible answers for each of these things. The ATP is the one that tends to throw people the most because they think they already know it. It's counterintuitive. So let's work on this step by step. In the relaxed state, if you go back to the previous slide, the relaxed state is about the 5 o'clock position on that circle. In the relaxed state, you see we have no calcium. There's no calcium to be found. So troponin and tropomyosin are covering up that high affinity binding site, that yellow patch, if you will. Myosin, on the other hand, is here. It's weakly associated. So one of the questions here that we would answer is we would say loose. It is loosely associated. It is not tightly bound to actin. The myosin head, this one right here, there are two of them, and it's not a good picture. I'd love to get rid of this lighter colored one. The myosin head here is in a cocked position. It is in a cocked position. How would we know that? It's hard to tell just looking at it, but we know it's cocked, and you know why? Because the ATP is hydrolyzed to ADP and PI. Listen very, very carefully. If this was not hydrolyzed, if all we had was ATP, the head would be bent over to the left. Okay? What we do with ATP is we shift from a flexed position to a cocked one. We are not generating pulling motion on actin by hydrolyzing ATP. We are setting up we are loading the myosin protein to release energy. So what you're doing is you're transferring that potential energy from this phosphate bond to the internal structure of the myosin. That's kind of like cocking. If you think about a flintlock rifle, if you know what I'm talking about, or even a regular pistol, have you ever seen somebody where they kick back the hammer, where they pull that hammer back and lock it into place? That's cocking. What you're doing is you're translating that energy into the hammer, and it's ready to go. It's not going to move. The potential energy is there. It's just that we've taken the potential energy and removed it from this bond and put it into the myosin head. Okay? That's what cocking is. We hydrolyze ATP to go from a flexed position to a cocked one. So we're in the relaxed state. We have loose binding. we have a cocked myosin head, and we have a hydrolyzed ATP that is now hydrolyzed to ADP and inorganic phosphate. Okay. So loose, the myosin head is cocked, and we have a hydrolyzed ATP. Okay, that's the relaxed state, and that's in the 5 o'clock position of that circle that we had in the previous slide. What we're going to do is we're going to change. We're going to move across the bottom of that figure. We're going to move across the bottom of that figure, and when that happens, that's the power stroke. So what happens? We get that second message that appears. We get that increase in cytosolic calcium. It binds troponin and tropomyosin. Binds troponin, and then troponin affects tropomyosin. Now that high affinity site is available. That high affinity site is available, and what happens? The first thing that happens is that we go from a loose affinity with actin to a tight one. So the myosin, and I'm going to give you a slapping sound, that's my hand grabbing my other wrist. We're going from a loose affinity to a tight one. And when that loose affinity shifts to tight, the first thing that happens is we lose the inorganic phosphate, and the head, let's look at this head here on the right. This head is in cocked position, and as this thing binds tightly to the actin, we lose the inorganic phosphate, and the whole head swivels into a flexed position. We did not hydrolyze ATP during this time. I just can't believe how many people don't get that. I don't understand that, because I try really hard to emphasize it, and I don't want to beat you over the head with it, but we do not hydrolyze ATP during this time. We bind tightly, the head begins to flex. As the head begins to flex, the first thing that pops off is the inorganic phosphate. So we increase the calcium binds troponin, the troponin pulls the tropomyosin off of that high affinity site, actin or myosin binds tightly, and it begins to flex. It begins to flex, and when it flexes, the first thing that pops off is the inorganic phosphate. This is going to be really important in the theory for fatigue. What we think is happening is there's a buildup of inorganic phosphate, and that prevents phosphate from leaving, and that prevents the myosin from completing its power stroke. But in any case, you don't see ATP hydrolysis during the power stroke. So the head is flexing. The first thing that happens is the inorganic phosphate leaves. And at the end of the flex, when we get to 5, the end of the flex, ADP will also leave. But it leaves in that order: inorganic phosphate first, and during the flex, and at the end of the flex, the ADP also leaves. This is the imparting of force on the actin. If there is ability to move the load, if there isn't too much load, the actin will get pulled in this direction. Okay. And this happens like you're walking your fingers across the surface of a table or something. Each head is alternatively cocking and flexing, imparting force on that actin filament. Be very careful with this. Work through this slowly. Calcium, troponin, tropomyosin. We bind tightly. One of the heads binds tightly. It begins to flex. It releases that inorganic phosphate during the flex, and at the end of the flex, the ADP leaves, generating force on that actin. And what we see is millions and millions of these myosin heads pulling against the actin, causing that shortening of the sarcomere. Here's just another picture of this. You see, in the relaxed state, we don't have troponin and tropomyosin here. There's no troponin and tropomyosin. In the relaxed state, the myosin head is cocked. The potential energy is stored inside the myosin right now, not inside the ADP. It's been hydrolyzed. We've already released that energy. Once the troponin and tropomyosin moves out of the way, we bind tightly. That binding, we begin to flex. The first thing we lose is the inorganic phosphate, and that head fully flexes, and what we don't see here is at the end of that flex, we lose the ADP as well. So after binding, the myosin changes. It goes from a cocked state to a flexed one. But we are not hydrolyzing ATP here. The ATP hydrolyzes to move the myosin back into the cocked state. Counterintuitive. Okay? So during the power stroke, first the PI detaches, the inorganic phosphate, and then the ADP detaches at the end of the power stroke. After the power stroke, the myosin will detach, but this is getting a little bit ahead of the game here. The myosin cannot detach unless there is another ATP to bind. So write that in there. Put a little asterisk right there, because the myosin will not detach unless ATP is present. So let's kind of work through this. We have here at the 5 o'clock position the relaxed state. What are the 3 questions? Tight or loose? The answer is it's relaxed, so it's loose association actin and myosin. Flexed or cocked? Well, we've hydrolyzed the ATP, so we are in the cocked position. If you can't tell from the diagram, by the way, here's a flexed one over here. You can compare it. By all means, do that. Use the figures. Use the figures to your advantage. And the ATP status, it's hydrolyzed ADP and PI, so we are not going to have hydrolysis from here to here. From here to here, there is no ATP hydrolysis. Okay? Where we see ATP hydrolysis is from here to here. From here to here. And we hydrolyze ATP in order to cock the myosin. We're flexed, we bind ATP, we hydrolyze it and cock the myosin. But from here to here, it's no ATP hydrolysis. So when calcium appears in the cytosol, the troponin and tropomyosin move out of the way. We get tight binding. We begin the power stroke. The myosin head begins to flex. As it flexes, the first thing that we lose is the inorganic phosphate. And at the end of the flex, we lose the ADP as well. And that brings us to the rigor state. Okay? So we get the calcium, we get the tight binding, we get that power stroke, we get the flexing going on, we lose inorganic phosphate, we lose ADP, and we cycle up here to the state. So we are now in the rigor state at the top, at the 12 o'clock position in that figure. And the answers to the 3 questions are, yes, we are tightly bound. The myosin is still tightly associated with the actin. The myosin head is flexed. Okay? This one should probably be flexed as well, and it's a little bit confusing. I'd love to get rid of this one. Maybe put a little white box over it or something. The myosin head is flexed, and you see there is no ATP, no ADP, no PI. There is nothing. That's the answer to the question in the rigor state. No ATP, no ADP, no PI, flexed and tightly bound. We need ATP to come in and allow us to release this myosin head. And that's what's happening. ATP comes in, it binds the myosin, and the myosin releases, first of all, releases the actin, and that allows it to hydrolyze the ATP. It hydrolyzes the ATP and it causes it to cock. So it hydrolyzes ATP and that cocks the myosin. We are not moving the actin at this point. We are simply going from rigor state, which would be the state in front of this, to relaxed state. We are transferring that potential energy into the myosin head, is what we're doing. So now we've gone from rigor state, which is not shown here. We bind ATP, we release myosin, we hydrolyze that ATP into ADP and PI, and we therefore cock the myosin head. You can see it's bound here, but we can infer that ATP, troponin and tropomyosin are there, and so it's binding weakly. And so we remain relaxed. The only reason we would continue contracting is if calcium is still present and the high affinity binding site is available. Again, a little bit of a tricky figure. Read the words as well as what you see in the pictures. So we've hydrolyzed that ATP. We are in the relaxed state, and if calcium is present, we'll go through another power stroke. If there is no calcium, the troponin and tropomyosin are covering those high affinity sites, and we will stay in the relaxed state. Okay. So the power stroke occurs when there's calcium present, causes the head to swivel, releasing PI, releasing ADP. We get to the rigor state. To get from the rigor state, we add more ATP. It binds the myosin. The myosin releases actin. That one head releases actin, and it hydrolyzes it and brings the head back into a Cox state. Again, transferring that potential energy to the myosin head. Now, later on in this chapter, we're going to talk about how we get from this down to this. This part right here is the excitation contraction coupling that we're going to talk about later on. But remember, we are not hydrolyzing ATP to swivel the head into the flexed position. We have already done that. So in the first part, as we begin to swivel, we lose the inorganic phosphate. At the end of the swivel, the end of that power stroke, we release the ADP. Now we're in the rigor state. And so that's how this works. You have to think about this dynamically. I promise you, I will ask a question about how do we get from rigor state to relaxed state? Or how do we get from relaxed state to rigor state? What's going on in the in-between? What is the dynamic process? Okay? What is the dynamic process? What occurs? What is changing between here and here in the three questions? What is changing between here and here in the three questions, or here and here? You should be able to describe the change between any and all of these panels. Guaranteed question on the exam. All right, that's the last slide today. Please, this is one of those big mechs. It's a dynamic situation. It's one of the tricky things in Chapter 12. Be ready for multiple questions about this on the exam, both multiple choice and or short answer. So thank you for your time today. I appreciate it. Good luck with all that you're dealing with the coronavirus, and I will talk to you next time. See ya.